Hi everyone, I'm Travis Sawyer. I'm an assistant professor of optical sciences at the University of Arizona, and it is my pleasure to have Dr. Jennifer Barton with me here today. Today we're doing an interview as part of the SPIE journal Biophotonics Discovery, which I'm an associate editor for, and for which Jennifer serves as a senior advisory editor. In this interview series, we're talking with leaders in our community to learn about their careers, gain insight from their wisdom, and understand what they foresee as some of the most exciting research topics on the horizon. So as a formal introduction, Dr. Jennifer Barton is a professor of biomedical engineering at the University of Arizona, the Thomas R. Brown Distinguished Chair in Bioengineering, and the director of the BIO5 Institute. She also has the distinction of serving as the SPIE president in 2024 and has numerous other awards and accolades. Now, I had the special honor of being one of Jennifer's PhD students who graduated back in 2021, and it is my pleasure to have her here with us today. Well, to start things off, I think our audience would love to hear a little bit about your background and how your career evolved. And so you're currently known as one of the leaders in biophotonics, so let's start off with, how did you get interested in this field in the first place?
Well, I've been interested in lasers since I can remember. In fact, I discovered in my parents' house not that long ago this fabulous report from my 13-year-old self called Lasers, where I predicted in 1979 that by the year 2000, lasers would carry all the telephone calls in the world. So what I knew at that point in time, and from experiments and things that were demonstrated on TV, was that lasers could be really helpful in healthcare. And that intrigued me. So I ended up pursuing kind of a circuitous path to get there. I studied electrical engineering as an undergraduate. Biomedical engineering didn't exist, and I'd never even heard of optical sciences. So, but eventually ended up getting my PhD in biomedical engineering, found a fabulous person at the University of Texas at Austin, A.J. Welch, who was working on laser therapies. So I ended up getting into that as a way of looking at whether or not laser treatment of the skin's blood vessels was working. We wanted a way of non-invasively imaging the skin. We thought if we could understand how blood vessels were configured in the skin, how large they were, how deep they were, we could potentially come up with individualized treatment, laser treatment for cutaneous disorders like port wine stains or hemangiomas. So that's how I got introduced to optical coherence tomography. That was a brand new technique at the time, and we thought, well, we just aim it at the skin and we'll get this beautiful map of blood vessels and we'll go from there with our models. That turned out to be a little harder problem than we thought, and I didn't quite get there during my PhD, although some really wonderful work has been done since, and now people are mapping the vasculature of the skin with great resolution. But that got me introduced into OCT, and I started looking at that as a way of not only looking at the skin, but skin cancer, and then other types of cancer throughout the GI system. Eventually ended up looking at it in ovarian cancer, and have been working on OCT combined with other optical modalities ever since.
That's excellent. I love hearing kind of the evolution of those research ideas and directions, and I think that probably many of our audience members understand how complex research is and that it's not quite as straightforward as you might originally think, but it's beautiful to see that kind of progression over time. Now, it was great hearing kind of your overview of research directions, and so I think one of my questions was, in general, for maybe some of our early career audience members, do you have insight or strategies that you would recommend to people who maybe are new faculty members starting their own independent research groups on how to develop those research directions that are complementary but also independent and that sort of thing?
Yeah, that's something that's always very interesting, and I was fortunate to have a mentor who was very generous with his time and ideas, and also towards the end of his career, I think he was very happy to have people take any and everything from his laboratory and move that out into their own program. Once I started working with OCT, I ended up going in a little bit different direction anyway. But I think, you know, I guess my advice would be to think about what really interests you at its most basic. Do you like developing hardware? Do you like image processing? Are you fascinated with working with other people? Would you really like to always be discovering new things, or do you want to settle in and become an expert in a narrow area? Those types of things are important because your opportunities take you in different directions. I would say the biggest mistake I made when I was a new faculty member is that I had this brand new technology called OCT, and it was very hot at the time. So I basically went around the University of Arizona, which had a brand new BME program. So I was all by myself, really with nobody working in my area, and said, "I've got this great tool. You know, I've got a great hammer. Show me a nail." And just kind of floundered into people and, asked them, "What do you think this could be useful for?" And so I tried all kinds of crazy things. So that was fun. But I think it was a lot of not taking the time to really understand the clinical problem, the biological problem, understanding really what it is that I wanted to do with the technology. I was just, going in 1,000 different directions at once. And I was warned about that. And it ended up being fine. The things that I found were it really is most important to me to be able to adapt technology for a really important clinical need. I actually am interested in all different kinds of things. So it's not like I wanted to only do cardiovascular or only do neuro or I find all of those fascinating. So what was important to me was finding the right collaborators. Since I knew I wanted to translate things into the clinic, I had to find physicians who were willing to work with me. So finding the right people to work with was really important, and I would absolutely not underestimate the challenge in finding good collaborators and finding good students. Take the time to do that right, because working with people who aren't a good match for you can lead to a lot of wasted time.
Excellent. That's terrific advice. Now, I'll come back to the idea of collaboration and especially physician collaboration later, but I do want to touch on your comments about mentors and specifically your PhD advisor, that we'd love to hear what are some of the people that you think of as the biggest mentors in your career. I'm sure he's one of them, but perhaps others as well?
Yes. So of course, my mentor, Dr. Welch, was amazing. I basically picked him off of a website because I knew that I wanted to do lasers in medicine. And so I found that I was, through circumstances kind of outside my control, was ending up in Austin. And so I looked up people, said, "Oh, look, here's somebody who does lasers in medicine," and just kind of stumbled into his lab. I got very fortunate. I mean, usually you can't do things that way, but fortunately, Dr. Welch was looking for students, and he appreciated that I had some industry experience, which I did at the time. And it worked out. And he was just, very kind and generous and forgiving. So one of the first things I did in his lab was break his one-of-a-kind laser.
Oh, our worst nightmare.
Yes. So he didn't kick me out of the lab over that, but we did have a discussion about how things work and maybe how to safely avoid breaking another laser in the future. But again, he was very important in my life. Other mentors, Jay Walsh is another faculty member at the time, was at Northwestern. He's since been to a couple of different places and had a couple of different roles. I ran into him again when we were both vice presidents for research. One of the best pieces of advice he gave me was when I was graduating, I was pretty sure I was going to go back into industry. After I got my PhD. I had come from industry and I knew I liked it. But then I started looking at these faculty positions and I was like, Jay, what should I do? Should I apply to faculty positions? Should I apply to industry positions? And he said, I think you could be happy at either. Why don't you just apply, and then if you have an opportunity, take a look at the details. So that was good because I applied here at the University of Arizona, and when I came to visit, I just knew it was a great fit. It was a brand new BME program. I was going to be starting something from scratch, had the opportunity to create something new. That doesn't come along very often, and so here I am.
That's awesome. Well, I, for one, am glad that you chose to take the academic route.
Thank you. So am I.
Now, you commented on holding a position as Vice President for Research, and I think that, a big part of your career has been these leadership efforts beyond what's kind of a standard faculty pathway. And so, at what point did you become interested in that kind of pathway where you can contribute to the field beyond research through different leadership efforts like that, or, of course, the SPIE President position?
Right. So I think more or less immediately was interested. And because of kind of the unique position I had here where we had a biomedical engineering division, it wasn't even a department at the time, and a graduate interdisciplinary program, everybody had to step up and help do some of the administration because there were only 5 of us. So I became involved in university leadership rather quickly, and then ended up actually taking over the division when the head left with another faculty. So now we're down to 3. When I had, that was actually the year I was supposed to take a sabbatical, and so I had just become an assistant professor and took over the department. But so that was kind of a unique situation where that ended up launching me into having administrative positions here, and I've had a number of them. But in terms of more related to research, I really encourage assistant professors, brand new people to, start small, start selective, but get engaged immediately. If you can serve as a trial reviewer, a junior reviewer, even for your own university's internal seed grant program, do that. Certainly if you can get on an NSF or an IH panel, there's some programs. Absolutely recommend that. It's an invaluable experience. Start reviewing papers. Of course, you have to do your fair share of those regardless, right? And I think SPIE is a wonderful organization, and so it's great to get involved. They do encourage junior faculty, junior people at all levels in industry, government, nonprofits as well, to get engaged in governance and in conference committees. So starting small, maybe asking to get on the conference committee for your own favorite conference, becoming a session chair. Those are little things that you can do to find out kind of what you like. Then from there, maybe you end up becoming a conference chair or getting onto a governance committee. Maybe you've always wanted to learn something about policy. SPIE has a policy committee that you can join for that. So to me, that's a really important part of being a faculty member or even just being a member of the optics and photonics community, because that's how you give back.
Absolutely. And I think that's especially important when there's kind of a lack of perhaps connection with the general public, that you may want to make sure that you understand where is that bridge between the public knowledge of what your science is and, of course, what our knowledge is. And so I appreciate that.
Yeah, that's absolutely a really important point. We all have a duty to explain the impact of what we do to the general public and to our elected officials. And it's easy to say, oh, somebody else can do that, or I'm not that good, I'm not going to do it, or yeah, people should just understand why we do is why what we do is important. And that's not the case. We have to explain. We have to explain our impact, explain why we should be getting federal funding, explain why what we do here isn't ivory tower stuff. It's actually important in making lives better for people.
Definitely, and I feel like of any field, optics is one of the ones with the most proximate impact to the public, since so much technology and healthcare and all of that is based on imaging and other optical technology.
That's right. That's right. Optics is everywhere. Lasers are going through fibers the thickness of a human hair and carrying all the weight. We don't use telephones anymore, but
that's funny.
all the videos and streaming in the world now.
Well, your prediction was right on that front. And so last question about background and that sort of thing, but I always think it's always interesting to have more of a personal aspect to these interviews. And so I'm just curious to learn about what are some of your favorite things to do in the copious amounts of free time that you have?
Yeah, I haven't had a whole lot of that lately. However, I do make sure to garden. I like to garden. I've moved to a small city lot, but just put in a whole bunch of raised beds and like to grow herbs because they grow well here in Tucson. I grow tomatoes because everybody needs to remain humble. Tomatoes definitely show me that I have no control over too much sun, too much heat, freak frost, bugs, everything that comes in. But no, seriously, mostly I enjoy that because it is very different than what I do every day. It's not in a laboratory, it's not in a computer, it's very, very tactile. And yet you get to see something grow, and even if you just get that one stunted tomato, you can say you grew it yourself. It's because of your own efforts, and that's very rewarding.
That's excellent. I also have recently cultivated an interest in gardening, but I have yet to have a successful tomato plant, so I understand the pain.
It's tough here.
Excellent. Well, thank you. So now I'm going to ask some more kind of research-specific questions. And in general, I think you're well known for pioneering some incredible miniaturized optical systems for different applications in biomedicine, and most prominently for the application of early detection of cancer like ovarian cancer. And so what would you say are some of the key advancements that enabled the development of these miniaturized technologies over the last 10, 20 years?
Yeah, it's all the advances in optics and photonics that have come out of academic research centers that have been commercialized by industry have made the work that I do possible. Also, scientific advances, I should say. I've been working on ovarian cancer for 15 years, and when we first started, we built a laparoscope. It was a big clunky device. It included large lenses and linear motors, and we built this rigid laparoscope, and we showed in the operating room that we could tell the difference between ovarian cancer and normal tissue. However, that kind of went nowhere, right? Nobody's going to go have a general surgery to get a diagnosis or, for a screening method. That's just ridiculous. So we actually didn't know what we were going to do there. But two things happened. First, science advanced, and they understood a little bit more about how ovarian cancer starts. It actually starts in the fallopian tubes. And we thought, oh, well, we could get to the fallopian tubes. You can get to the uterus pretty easily, so we ought to be able to get into the fallopian tubes. Oh, but they're a millimeter in diameter. So we kind of had to wait for fiber optics, for better materials that have pushability and lubricity, for tiny, tiny fiber guides to come up, for OCT acquisition to get faster. Once all those things caught up, all of a sudden we were like, well, we can do that now. Wow. So it was pretty exciting to actually be able to build an 800 micron diameter dual modality optical endoscope that could go into the fallopian tubes. And I'm just excited about where technology is going to lead us next. Absolutely. That's really exciting. And it's amazing to kind of be on the cutting edge. And also it speaks to the interdisciplinary nature of this type of work, that you're integrating concepts in medicine, in basic science, in materials, into this really impactful product. Yes. Now, I understand that you are now conducting clinical trials in patients, which I think is amazing and extremely exciting. But what are some of the biggest challenges at this stage of the research? Yes, yeah. So first of all, they're clinical studies. We're not quite to the trial stage, and the reason is that gets to be extraordinarily expensive. So we can do studies. We can demonstrate proof of principle. We can prove out and refine the technology. But once you want to get to the stage of actually proving that this technology not only works, but that it can save lives and that it can cost effectively save lives, gets to be something that starts now not costing $1 million, but 10 or multiple tens of millions of dollars. And so at that point in time, you need an industry partner, probably some venture funding to take that on. Makes sense. So that's the stage I'm at now. Ovarian cancer is a somewhat rare disease, and so finding somebody, there is a business model for it. We've been through that, but finding somebody who's interested in taking on that risk for what is, could be a commercially successful product, but is also going to be a labor of love is where we are now. So we're talking with people and learning all types of things that I didn't learn as a graduate student, like, how do you come up with the cost of materials? How do you do things in bulk? What's the regulatory pathway? Those are all the things we're dealing with now.
Interesting. I think it's fascinating how all of these fields are so deep that you can get trained in something for 15 or 20 years and then know absolutely nothing about a topic, which is actually not too far from what you have been working on. That's excellent. But now I want to talk about a few of the publications you've had in Biophotonics Discovery over the last couple of years. And so specifically there are two. And first I wanted to ask you about your publication focused on random sampling of second harmonic generation images for colon cancer. And this was published and featured on the journal cover. And so I wanted to ask you, could you comment on how this innovation, the innovation laid out in this paper, could improve our management of colon cancer in general?
Yes, so the idea there was that if we want to make, speaking of cost-effective devices, let's make them as simple as possible. So if we want to do, first of all, multiphoton microscopy is a technology I thought, 15 years ago would never end up in the clinic, right? Because you had to have this huge laser and all these difficult optics and things. And actually now you can get a shoebox laser and some simple fibers and other items like that and put together an endoscope. But one part that has remained kind of stubbornly difficult, large, and expensive is scanning. And with you probably want to do that at the end of your endoscope, so that kind of limits how small you can make it. So the thought was, well, we know that the signal from cancerous areas is different than the signal from normal areas. Do we actually have to create an image? We're used to looking at that spatial relationships, and there's been great work done, and we've shown that fiber thickness and density. But can we, if we just randomly take measurements at a certain depth in tissue, can we tell just from that? And this paper was proved that we could do that. We actually took the images themselves and then said, okay, well, we know we have truth from the images. If we just kind of randomly take pixels from that image, can we get to the same conclusion? And it turned out that by just getting about 1,000 samples, you can.
Interesting. Seems like a very elegant and straightforward solution to, as some of us know, how complex scanning is, especially in something like a miniature endoscope. So a very interesting publication. And now your second publication, which was published in May 2024, focused on using OCT to detect electrothermal damage in the ureter. And I thought that this study was a great collaboration with physicians and scientists aimed at addressing a really clear unmet need. And so I was wondering, could you comment on how this research direction was originally formed? And then second, what are the best practices for forming such collaborations and most efficiently running them?
So yeah, those are great questions. So this came about because the gynecological surgeon that we work with on our ovarian cancer research started, thinking about all the other problems he had, and so he's great. And that's a really nice way to get into new areas, is to talk with a proven collaborator about maybe other problems they had. And one of the things that he said is that gynecological surgery is very, very difficult. You're working with a whole bunch of connective tissue in which are embedded highly important things like ureters, and you can't really see them. So you do your best and to make sure that when you're doing something like a hysterectomy, you don't, where you're using thermal coagulation to cut the tissue, that you don't get near the ureters. But because patient anatomy can change and can be very different from one patient to the other, occasionally you do accidentally thermally coagulate a ureter. Now, it's standard of care that at the end of a procedure like that, you put a cystoscope in the bladder and you look for urine flow. Well, that will tell you didn't cut the ureter. But if you damaged it, you won't realize until about 3 days later, when the tissue necroses and you start having leakage, that there's a problem, at which point in time you got to bring the patient back and put in a stent until the ureter heals. So we had an opportunity here to say, well, we're already going into the bladder. If we put a probe up into the ureter, could we detect whether or not there had been any damage? So this was the first proof of principle study. We did it in pig ureters because that was a good way of proving whether or not the idea would work, whether or not OCT could actually see thermal coagulation. It can very well. So next step would be to move on to a human study. And you're saying, well, how do you have a human study where you're trying to look at damage when your whole point is that you don't want to damage? So, this is where really interesting discussions with your clinical collaborators can come up about how could we make something like this happen? Well, he knows other people in urology who are doing things like taking out non-functional kidneys with ureters. So the next step will be to have a study where you have a patient who's getting their kidneys and ureters removed anyway, and it will be much more straightforward to obtain consent to be able to do a little test prior to removal where you intentionally damage.
Interesting.
And see whether or not the types of damage that might occur during a surgery can be visible.
Interesting. That's a very clever solution. I think that we see similarities in things like esophageal cancer screening, where the tough part is finding the early cancer, but you don't know when it's there, so how do you collect samples prospectively?
Exactly.
But that's amazing, and I feel like that clinical collaborator is like a dream come true. They come to you with the problems to use your technology instead of having to go search for them.
Right. And I think one of the keys with clinical collaborators is to realize that they're like, you can have acquaintances and you can have best friends, right? It's not an all or nothing type of thing. There are physicians out there who really don't have the time or maybe the motivation to do much with you, but they're happy to give you samples or give you advice or talk to you, every couple of months. And those are great. But you really do need to find one who's dedicated and passionate about research. It is hard to incorporate research into a busy clinical practice, and you may only come up with one or two of those, but those are the people who you really treasure.
That's excellent insight.
Yeah.
Thank you. So now I'll kind of transition to some closing questions. And as someone who wears many hats and somehow accomplishes a volume of work equal to several people, do you have to make compromises in what you undertake? And what is your decision process in doing that if you do have to make such a compromise?
This is one of the toughest things, right? There's an opportunity cost to everything that you do. There's only so many hours in the day. And I think it goes back to something that I hope I've conveyed through this, which is, really think about what your passion is, what you want your purpose to be, what you want your goals to be in life. Everything I do is about building. I love to build. I love to build endoscopes. I love to build careers of students. I love to build programs, the biomedical engineering program built here. I love to build programs here as director of the BIO5 Institute. I love to build relationships through being president of SPIE. And so when I have an opportunity, I look at it through the lens of that. Is this really something that I'm passionate about doing? Is this aligned with my core purpose? Am I really the best person to do this? And I wish I could say I made the right decision all the time. I don't. I frequently end up doing things and I'm like, why am I doing this? But I've gotten better at that over the years by kind of intentionally thinking, not saying yes immediately. And then there's also things that you just need to do because relationships are the most important. And so if it is about helping, assisting, building a junior person's career, about helping them get ahead, that's definitely my highest priority.
That's amazing. A true engineer, always building.
That's right.
And so speaking of early career, how about new students? When you're talking to new students who are interested in biomedical optics and want to join this research community, do you have thoughts or advice on how they can best position themselves to pursue this and make an impact?
Yeah, so students, that's the best time of life, right? Being a graduate student, particularly a graduate student post-comp exams, It's like the best time ever. So, what do you tell a new student coming in? It's kind of the same thing where, what's your passion? What's your purpose? What do you want to accomplish? Of course, I was completely wrong about what I was going to do when I graduated, so I don't usually ask that question as much. But what skills would you like to have by the time you graduate is maybe another question. Putting together an individual development plan is important, whether that be formal or just simply thinking regularly about, okay, I've got a certain period of time, how can I accomplish what it is that I want to get done by the end. I also tell students that really there's a lot of things you can do, here at the University of Arizona and frankly at every university. There's so much exciting stuff going on. You could be happy doing, dozen different projects. Where's a laboratory that you feel you fit in with a culture is really a much, much more important question, where you get the type of mentoring that you need, the frequency of meetings that you need, whether it's very individual-based or team-based. Those types of questions are important to ask, and be honest with yourself about whether or not this is really a laboratory that you feel good being in and you feel you can prosper.
I really appreciate that comment. I think that it seems like for many students, the focus is always the research topic and what is the right choice. But I think if I'm interpreting correctly, you're saying there's many good choices on the research front, and to also pay attention to some of these other factors, like how do you feel in the community and the support that you get. That's excellent advice. So now the last question for today, what is it that you're most excited about in the next, let's say, 10 years of biophotonics?
Oh gosh, well, I feel like biophotonics has, it's past its childhood, it's even past its kind of awkward adolescent phase. We're now becoming a fully fledged adult, and that's the time when these advanced imaging modalities are really finding their place. They're not a panacea for everything. They are being adapted for very specific needs and making a real difference in clinical practice and in making lives better. So that's what I'm excited about, is seeing the stage where things move into commercialization, become lower cost through greater volume and technology improvements, and just become established the way that, I'm always jealous of CT and MRI as being such well-established imaging modalities. I'd like to see optics get there as well.
Fantastic. Well, thank you again, Jennifer. Thank you so much for your time. And thank you everyone for listening. Please stay tuned for other interviews in this series, who will be held by other members of the Biophotonics Discovery editorial board, and we'll be interviewing other incredible leaders in our field like Jennifer. It'll be terrific to hear their insights and perspective like we heard from Jennifer today. So thank you again.